Very good morning, students. Myself, M. Pradeep, lecturer in biotechnology, and today we are going to discussing about cell division, types, and significances. What is cell? You all know cell is the basic unit of all living organisms, starting from prokaryotes, that is unicellular organisms, to higher organisms like humans. All these living organisms made up with cells. So cell is the basic unit of living organisms, and a cell is a dynamic entity, not a static entity. That means cell always trying to divide and produce more and more cells. Because of that reason of or cell division nature of cell, the organism increase its size and also differentiated into different types of cells, organs, and tissues. So, what are the different types of cell divisions? There are generally two types of cell divisions exist in eukaryotes. Those are mitosis and meiosis. These are cell divisions mostly observed in higher organisms. Like humans, we have these two types of cell division. First, lower organisms like bacteria, protozoa, they have simple divisions called binary fission. But we have two types of cell division. Those are mitosis and meiosis. If you take eukaryotic cell, generally the cells are categorized into two types. Those are prokaryotic and eukaryotic cells. Prokaryotic cells doesn't have any nucleus, whereas eukaryotic cells contain nucleus. And if you take eukaryotic cell grows and the DNA replicated. These two things observed in the eukaryotic cell. That is one cell growth and the second one is DNA replication. Mitotic cell division produce the daughter cells identical to the parents. As I said, there are two different types of cell divisions there. One is mitosis and another one is meiosis. In mitosis, one cell produce two daughter cells. And parent cell produce two daughter cells, and these two daughter cells are identical in nature. That means their chromosome number, their shape, and everything is identical. And this is different from prokaryotic cell division. That is, in prokaryotes like bacteria, the cell division is binary fission. That means one cell divide simply and produce two bacterial cells. Whereas mitosis also similar procedure, but is it different from the prokaryotic cell? Because in prokaryotic in mitotic division there are two phases are there. One is interphase and another one is division phase. If we see here this cycle, G1, S and G2, these three phases are called interphase, and this M phase is known as division phase. After the division phase, one cell become produce two daughter cells. So this is mitosis occur in the eukaryotic cells. But in prokaryotes, the binary fission doesn't possess this type of interphase and division phase distinction. So that's why the eukaryotic mitosis is different from prokaryotic cell. Eukaryotes have more DNA on many linear chromosomes. And another thing is prokaryotes have, as I said earlier, the prokaryotes have circular DNA, whereas in eukaryotes there are linear DNA present. How many number of chromosomes present? That much number of DNA molecules present in the uh, eukaryotic cell. For example, humans, if we take humans, the replied cell contain 46 chromosomes. So 46 number of DNA molecules are present in that. Whereas in prokaryotes like bacteria, only one DNA molecule present, and that too circular in nature, not linear nature. The timing of replication cell division is highly regulated. In eukaryotes like humans, the replication is restricted to particular time only. That is here there is S phase is there in the cycle G1, S, G2. Here S phase is there. In this S phase only DNA replication taking place. But in prokaryotes, entire process of binary fissions, DNA replication taking place. So this is the distinction between prokaryotic cell and eukaryotic cell during mitosis. In eukaryotic cell cycle, there are mitosis. There are two major phases are there. One is interphase. G1, S, and G2 phases are called interphase, and M phase is known as division phase. So these two phases are called interphase and division phase. The interphase is preparative phase. That means if you want to write an exam, the exam only conduct in 1 or 2, 3 hours only, but we have to prepare throughout the year. So here also the interphase is long, whereas the division phase is very small. So the interphase is the preparative phase. That means what for preparation? Division. So it prepares for division. So this much long time taking for division. So this is preparative phase, that is interphase, and this is division phase. These are the two major phases of eukaryotic cell during mitosis. If you take interphase, it's a non-dividing state with the three substages. Interphase is, as I said already, interphase is the preparative phase. It has three subdivisions, subcategories. Those are G1 phase, S phase, and G2 phase. In G1 phase, cell grows in size organelles replicated. In this G1 phase, cell size grows, organelles also increase. Cell organelles, there are several cell organelles, mitochondria, chloroplast, for example, plant if you take, and another endoplasmic reticulum, Golgi complex, lysosome. These are all cell organelles. All these cell organelles number increases and the cell size also grows. In the second stage of interphase is S phase, where DNA replication taking place and one DNA molecule produce two DNA molecules. And the third phase is G2 phase, synthesize the proteins associated with the mitosis. Immediate to the G2 phase, there is M phase is there. For M phase or division phase, whatever the proteins required, that proteins is synthesized in the G2 phase. So in this way, interphase has three different subphases. Those are G1 phase, S phase, and G2 phase. Then after interphase, there is a actual division phase taking place. This one, this is the division phase or M phase or mitosis phase. Division of somatic cell. Somatic cell means non-reproductive cells. In eukaryotic organism, a single cell divided into identical daughter cells. So daughter cells have the same chromosome number the parent cell have. This is the parent cell. It has one mother chromosome and father chromosome, and DNA replication taking place in S phase, and these separate then these undergo meiosis, division phase, then produce two daughter cells. So in this way, meiosis after interphase, then undergo division phase and produce two identical daughter cells that are similar to the parent. This is parent one, this is the daughter cells. These two daughter cells are very much similar to the parent cell. That's why the mitosis is also called equivalent division. That means it produces two daughter cells that are similar in nature. Packing from the move. When cell is not dividing, if cell is in undividing state, that means in resting state, DNA molecules in extended uncondensed form, that is chromatin. So this is called chromatin. So it is uncondensed form. DNA is loosely packed when it is in resting stage. Cell can only replicate and transcribe DNA when it extended state. Then in this loose form, these DNA molecules produce another DNA molecule by replication process. So this DNA molecule become two DNA molecules. After that production of another DNA molecule, then it go for condensation. Condensation means converted into chromosome. When cell is preparing for division, DNA molecule condenses to form chromosome prior to the division. So each chromosome is single molecule of DNA. Each chromosome has one DNA molecule, and easier to sort and organize the duplicate DNA into daughter cells. So whenever DNA is in the cell is in resting state, the DNA is unpacked state. Unpacked means it is loosely extended state. Then it undergo replication, produce another DNA molecule, then it start condensed and produce chromosomes. After chromosome formation taking place, then only cell division takes place. So this mitosis division phase has four phases again. As I said, mitosis contain generally occur in two phases. One is interphase, another one is division phase. So interphase has three subphases, G1, S, G2. This is called preparative phase. After preparative phase or interphase, then undergo division phase. In this division phase, again four phases are there. Those are first one prophase, second one metaphase, third one anaphase, and fourth one telophase. So these are four subphases of division phase. This is followed by cytokinase, that means cytoplasmic division. So mitosis cycle, if you take mitosis cycle, cell is in resting state, undergo preparative phase, then it produce interphase, interphase. This cell is in resting state. Whenever cell want to undergo division, it go interphase G1, S, G2 phase, then division phase. This is prophase, metaphase, anaphase, and telophase. After telophase, the cytokinesis takes place and produce two data cell. Again, one of the cell undergo cell cycle, and again DG1, S, G2, prophase, metaphase, anaphase, telophase. Like this cell division taking place. What happens in the prophase of cell division phase? Major events. What are the major events? Chromosomes condensed. In this, as I said earlier, whenever cell is in resting phase, the DNA is in loose form or extended form. Whenever cell want to s- divide or undergo cell cycle, then the loosely packed DNA become condensed and form chromosomes. And these chromosomes are attached with the spindle fibers, and these spindle fibers separate the chromosomes to two daughter cells. Chromosomes are captured by spindles. Here, these are the spindle fibers, these blue color ones. These are the chromosomes, and these chromosomes are captured, attached to the spindle fibers and separated apart. So, this is the prophase stages. This is the prophase: chromatin, nucleus, nucleolus, and these are the centrioles. These centrioles form the spindle fibers, and these spindle fibers attach to the chromosomes at the centromere and the cells, separate the chromosomes. So this is prophase. Then metaphase. In metaphase, all the chromosomes attached to the mid plate, that is one plate. So chromosomes along equator of the cell with the one kinetochore facing each pole. They are facing one of the each. This is one pole and this is one pole. They are separated by each other during metaphase. So metaphase is the one of the best stage to observe the chromosomes in a cell. So if you want to observe the chromosomes in a cell, which stage is the best stage is metaphase. Then anaphase, followed by the metaphase, anaphase is there, where sister chromatids are separated. Sister chromatid means each chromosome contain two chromatids. One chromatid goes in one way and another chromatid goes another way. In this way, this is one sister chromatid, this is another sister chromatid. These pulled apart by these spindle fibers and separated. So in anaphase, the chromosome sister chromatids are separated. Here, this is the actual picture of the anaphase showing microscopically. So, free spindle fibers lengthen and push pulls of the cells apart. Cells become that chromosomes become separated in anaphase, whereas in telophase, spindle fibers they disintegrated. There is no need of spindle fibers in telophase. Then the separated chromosomes surrounded by the nucleus and waiting for the cytokinesis, enclosing each daughter nucleus into separate cell. This is midplate, and this separate these separated nucleus into two different cells. So cytokinesis, plant versus animal cell. In plant cells undergo cytokinesis by forming a cell plate between two daughter cells. This is cell plate. This is separated chromosomes. These separated chromosomes surrounded by the nucleus, and the mid plate separate these two nucleus as a separate cells. Whereas in plant cells, whereas in animal cells undergo cytokinesis. That means this cell undergo furrows. That means the cell membrane integrates into invaginate into towards nucleus through the formation of cleavage furrow. This is called cleavage furrow. This is entire cell. This plasma membrane undergo furrows. A ring microtubule contract pinch the cell into half. These two cells separated as apart furrow extended, and these two cells are separated. This is in case of animal cell. There is no midplate formation in animal cells. So the stages of mitosis, if we take, there are interphase, and a division phase. In interphase, there are G1, S, and G2, whereas in division phase, prophase, metaphase, and anaphase, and telophases are there. This is mitosis. Genetic terminology. If you take another type of cell division is also there, that is meiosis. If you take genetic terminology, sexually reproducing eukaryotes have two types of body cells. One is somatic cells, as I said earlier, and these somatic cells undergo mitosis. Another type of cells called sexual cells are allosomes. Sexual cells are sex chromosomes, somatic cells and reproductive cells. Another type of cells are sex cells. These produce gametes. For example, if you take a somatic cell and this is sex cell, somatic cells undergo mitosis, whereas sex cells are reproductive cells, also called. They undergo uh, they undergo meiosis. Another type of cell division called meiosis. What is cell division of gametes called meiosis? So in meiosis process, gametes produced, whereas in mitosis, similar type of cells produced. So a single germ cell or reproductive cell or sex cell divides into 4 unique daughter cells, whereas in mitosis there is only 2 daughter cells produced. But in case of meiosis, there are 4 daughter cells are produced, but all these daughter cells are haploid in nature. In mitosis, the daughter cells also have diploid in nature, whereas in meiosis, the daughter cells produced are 4 and they are in haploid in nature. Daughter cells half of the chromosomes as parent cell, so they consider haploid. If you take this thing, these two are homologous chromosome. These are homologous chromosomes. Interphase division takes place and to produce and the chromosome DNA replication taking place and homologous recombination takes. Finally, it produces 4 daughter cells and 4 daughter cells are different from the parents. And these are 4 chromosomes are there, but in daughter cell only 2 chromosomes are there. So half of the number of chromosomes present. So haploid in nature. Genetic terminology ploidy. What is ploidy? Refers to the number of sets of chromosomes in a cell. Haploid means one set of chromosomes. For example, if human 46 chromosomes mean 23 sets, so two sets of 23 chromosomes. So it is diploid. If any human cell has only one set of chromosomes, that is 23 chromosomes, that is called haploid. So all sperm and eggs are haploid in nature, whereas other all somatic cells are diploid in nature. Diploid organisms received one set of each type of chromosome from female parent and another set is from the male parent. We have 46 chromosome in each and every cell of the nucleus. And these 46 chromosomes, how the cells get means we get 23 chromosomes from father and 23 chromosomes from mother. So we are diploid. Homologous. What is homologous? Chromosomes exist in homologous pair in diploid. But in each and every cell has 46 chromosomes. These 46 chromosomes are two sets of 23 chromosomes, and each chromosome has in two copies. For example, the first chromosome has 2 copies. The second chromosome has 2 copies. Third chromosome has 3 copies. Like that, we have 23 pairs of chromosomes. In each pair, one chromosome coming from father and another chromosome coming from mother. And these father and mother chromosome form homologous pair. So this is, for example, we take first chromosome. This is father chromosome, this is mother chromosome, and this first father chromosome and this first chromosome, first mother chromosome combined form homologous pair. So sex chromosomes, if you take these all 22 autosomes, we have this type of situation chromosome. But whereas in sex chromosome, in case of male, we have X and Y chromosomes, whereas in females, XX chromosomes are present. So except sex chromosome, remaining all autosomes we have pairing. Other chromosomes known as autosomes, they have homologous pairing. If you take karyotype means whatever the chromosomes present in a cell, they are arranged in a particular order, either ascending or descending order, that is known as karyotype. This karyotype is very much useful for the identification of a disease condition or integrity of the chromosomes in a cell. So based on that, we can know that whether any genetic defects are there, birth defects are there, or any alterations are there in the chromosome structure or nature, we can identify. So in this way, karyotype is very much useful for the identification of normal person or diseased person. So asexual reproduction, what is asexual reproduction? Whenever only one parent is involved in sexual reproduction, that is called asexual reproduction. Whereas two partners are involved, that is sexual reproduction. Asexual reproduction means many single-celled organisms reproduce by splitting or budding. So those are called asexual reproduction. Some multicellular organisms can produce asexually produce clones. So, example, Hydra. Hydra it also produce asexually reproduce. Sexual reproduction. Fusion of two gametes to produce a single zygote. For example, female, male. Female produce eggs and male produce sperms. These eggs and males combine to form zygote. Introduces greater genetic variation allows genetic recombination. And in sexual reproduction, two parents involved, one male parent and female parent. Male produce eggs and male produce sperms and female produce eggs. And these two fuse and form zygote. And genetic variations also occur in the sexual reproduction. With the exception of self-fertilization organism, zygote has gametes from two different parents. So in sexual reproduction, in case of humans, we have two genetic information on two partners. Sexual reproduction in humans. At fertilization, 23 chromosomes are donated by each parent. And that means during fertilization, egg contain 23 chromosomes, which are coming from mother, and sperm contain 23 chromosomes coming from father. This is egg, this large one, and they contain 23 chromosomes. And this is sperm, it also contain 23 chromosomes. These two fuse and form called fertilization. This fertilization 23 plus 23, 46 chromosome zygote formed. Gametes contain 22 autosomes and one sex chromosome. These are haploid. Fertilization result, these 23 egg and 23 chromosome containing sperm combine to form 46 chromosome containing zygote. Most cells in the body are produced through what type of cell division? In our body, most of the cells undergo mitosis only, but only small part of reproductive cells are there, that is in gametes. For example, in case of female, that is ovaries are there, whereas in males, that is testes. These two structures in humans undergo meiosis and produce gametes. The remaining all the body cells undergo mitosis only. Meiosis is also called sexual reproduction. In meiosis, there are two divisions of the nucleus, meiosis 1 and meiosis 2. Whereas in mitosis, only one interphase, one division phase is there. But in case of meiosis, one interphase, two division phases are there. These division phases are categorized into meiosis 1 and meiosis 2. Here we take this is interphase cell. This undergo interphase DNA replication S phase and undergo G2 phase, then undergo prophase 1. That means meiosis 1. Meiosis 1 again, four subphases, prophase 1, metaphase 1, anaphase 1, telophase 1. In this anaphase, only homologous chromosomes pair separated. That means father chromosome and mother chromosome separated. In meiosis 1, contain anaphase 1, the father and mother chromosomes are separated. That is homologous pair chromosome separation taking place. That's why in meiosis 1, the diploid cell become haploid. Then meiosis 2, anaphase 2. In meiosis 2, there is anaphase 2 contained. In this is actual mitosis. This is similar to mitosis. The meiosis 2 is similar to mitosis where chromatid separation takes place. That means during meiosis, one interphase is there. Interphase is followed by division phase. This division phase contain two divisions. One is meiosis 1, another one is meiosis 2. In meiosis 1, there is anaphase 1 is there. In this anaphase 1, chromosomes separated. What chromosomes? Homologous chromosome. That means father chromosome, mother chromosome separated. That's why the diploid cell become haploid. So in meiosis 1 only, anaphase 1, the diploid cell become haploid. Whereas in meiosis 2, that is in anaphase 2, only the chromatid separated. Chromatid separated, which is similar to mitosis. In this way, cell division taking place and produce four daughter cells and are haploid in nature. So meiosis and sexual reproduction, diploid organism. Ovaries, testis. As I said earlier, these two locations only in humans, meiosis take place. In case of females, ovaries, whereas in males, testis. Ovaries undergo meiosis and produce gametes called eggs, which are in haploid nature. Testis also undergo meiosis and produce gametes called sperms. These gametes and eggs and sperms fuse fertilization and form zygote, and these zygote undergo mitosis, produce diploid organism. In this way, meiosis help in the production of sexual gametes, and these gametes fuse and form zygote and produce whole organism. Genetic variation, as I said earlier, in meiosis, genetic variations is the cause. Because why, how genetic variation cause means fusion of sperm and egg result in unique offsprings, but not only because the young are product of two individuals with different genetic makeup. Meiosis shuffles the genes so that individuals' gametes are genetically different from each other. In anaphase, there is a chromosome separation, a chromosomes mixing, a chromosome exchanging taking place, that is called crossing over. So in this crossing over, genetic exchanges taking place. Because of these genetic exchange or genetic modifications, the variations also taking place. Genetic shuffling meiosis 1, crossing over in prophase 1, independent assortment in metaphase 1. So these two processes in meiosis 1 responsible for the genetic variation in meiosis. What is crossing over? Here we take, this is one father chromosome, this is mother chromosome. These two chromosomes called homologous chromosome. They form X-shaped structures in their chromatid called chiasmata. In chiasmata, they are exchanging of chromosome. That means blue color one get orange one and orange one get blue one. In this way, genetic exchange taking place. This genetic exchange causes variations in the offspring. Independent assignment. What is independent assignment? Whatever the chromosome coming from father and whatever the chromosome coming from mother, they as it is, they are not going to another cell. So one father chromosome and another mother chromosome enter into cell. This is assortment, but it is independent assignment. We take this is father, this is mother, this is father, this is father. We shall get one father chromosome, one mother chromosome, father chromosome, mother combination of chromosomes will taking place. Because of this combination, that means father, some of the father chromosome enter into one gamete and some of the mother chromosome enter into one gamete. So mixing of these chromosomes during cell division taking place, this is called independent assortment. That's why the variations also taking place. Spermatogenes. What is sperm? Males produce sperms through the life. After the onset of puberty, about 1500 sperms per second produce. Spermatogenesis. This is testis cell, diploid cell, undergo division. These divisions finally produce sperms. This is called spermatozoa, and this process is known as spermatogenesis. Oogenesis. In spermatogenesis, all the four sperms are active. That means, as I said earlier, whenever cell undergo meiosis, produces four cells. All the four cells are active in case of males as sperms. Whereas in eugenesis, which is occurred in females' ovaries, after on a primary oocyte, this is called primary oocyte, which is present in diploid and oocyte, and this undergo uh, meiosis 1 and meiosis 2 finally produce 4 cells, but only one mature ovum is active, remaining 3 undergo become polar bodies. So in this way, oogenesis, only one cell is active. Mitosis and meiosis. In mitosis, differences means mitosis, the daughter cells are diploid in nature, whereas meiosis, haploid in nature. These are called clones. In case of mitosis, the daughter cells are called clones, but in meiosis, the daughter cell different from the parent cell, as parent cells are diploid in nature, whereas daughter cells are haploid in nature. Same genetic information carried out in meiosis, but here exchange of chromosomes technically that new characters come. Give me another one just like the other one. Same type of individuals form, cells are formed. A shuffling of the genes, see, and different variations taking place in this. Yes. If you draw a chromosome, this is resting cell stage. Whenever it undergo interphase and S phase, it produces two chromatids like this. This is unreplicated condition. This is replicated condition undergo divisions. So in this way, this is all about cell division, which are categorized into two types. One is mitosis, another one is meiosis. Mitosis occur in the parent cell, somatic cells, whereas meiosis occur in the uh, reproductive cells. Where reproductive cells are present means in case of female that is present in ovaries, whereas in males it is present in testes. So this is about cell divisions and the types. Thank you for listening.